There's a forest somewhere in Europe that was supposed to be dead. The trees had stopped breathing, the soil had gone silent, the birds had left. Scientists stood at its edge with clipboards and grim faces, writing conclusions that felt more like obituaries. Every conventional method had failed, every intervention had backfired, and then someone proposed something so counterintuitive, so scientifically audacious, that half the research team thought it was a mistake. They didn't plant new trees, they didn't spray chemicals, they released insects, 10,000 beetles into a forest already dying. What followed rewrote everything we thought we knew about life, death, and the hidden intelligence of nature. Nobody told you that forests can scream, not in the way you and I do, not with sound, not with movement, but in the language of biology. A dying forest broadcasts distress signals so precise, so layered, so chemically complex, that the atmosphere around it literally changes composition. The trees release volatile compounds into the air. The soil bacteria alter their metabolic rhythms. The root networks those underground communication systems that scientists have only recently begun to decode pulse with what can only be described as emergency signals. And in the middle of one such dying forest, a team of ecologists made a decision that the scientific establishment was not prepared for. They did not bring medicine. They did not bring machinery. They brought beetles, 10,000 of them. And what happened next didn't just save a forest. It forced the entire field of conservation biology to confront an uncomfortable truth it had been avoiding for decades. To understand why this matters, you need to understand what a dying forest actually looks like from the inside, not from the outside, where it might simply appear brown, sparse, defeated. From the inside, a forest in ecological collapse is a study in cascading failure. Forest disturbances such as insect pest outbreaks, fires, and wind throws have a substantial effect on the functioning of these ecosystems, rippling through their carbon storage, their primary production, and the invisible microbial architecture that holds everything together. But here's what most people are not seeing. The collapse doesn't happen all at once. It happens in a sequence. First the canopy thins, then the light patterns on the forest floor shift, then the fungi respond, then the soil bacteria, then the birds, then the larger animals. By the time a forest looks dead to human eyes, it has already been dying at the microscopic level for years, maybe decades. The tragedy is not the death itself. The tragedy is that we consistently arrive too late, misread the signals, and apply the wrong solutions. This particular forest had been through it all. Logging operations in the mid-20th century had stripped it of old-growth complexity. Monoculture replanting had replaced the original mixed species ecosystem, with rows of Norway spruce orderly, economically efficient, and ecologically brittle. Compared to healthy forests, where deadwood may represent only about 5% of above-ground biomass, forests in distress can see deadwood rise to represent up to 80% of their above-ground biomass, creating conditions that fundamentally alter how nutrients move, how carbon is stored, and how every organism from bacteria to birds finds what it needs to survive. 80%. Think about what that number means. It means the forest had become, in biological terms, a graveyard pretending to be a woodland. And into that graveyard, the researchers had been throwing everything the textbooks recommended. Controlled burns in some sectors, selective harvesting in others, soil amendments, seedling transplants. Nothing worked. Or more precisely, nothing worked fast enough, and nothing worked at the right scale, and nothing addressed the core problem that everyone in the room could see, but nobody wanted to say out loud. The core problem was absence. Not the absence of trees. There were trees, damaged and stressed, but still standing. Not the absence of water rainfall in the region had actually increased slightly over the study period. The core problem was the absence of the organisms that make a forest function as a living system rather than a collection of individual plants. And chief among those missing organisms were beetles. Specifically, a class of insects known as saproxylic beetles, wood-dependent species that spend their life cycles inside dead and dying wood, breaking it down, creating microhabitats, inoculating it with specific fungal species, and setting in motion a chain of biological events so complex and so interconnected that it had taken decades of research just to map its outlines. Deadwood provides the substrate for a multitude of wood-dependent organisms, as well as a variety of microhabitats, and deadwood is a key factor in maintaining ecosystem functionality and increasing overall forest biodiversity. Without these beetles, the deadwood wasn't decomposing properly. Without proper decomposition, the nutrients locked inside those fallen trunks and dying branches weren't being released back into the soil. Without those nutrients, the surviving trees couldn't mount effective defenses. Without effective defenses, they were vulnerable to stress, to drought, to pathogen attack. The beetles weren't decoration, they were the engine. But here's where it gets worse. The absence of saproxylic beetles wasn't just a local problem in this particular forest. It was, and remains, a systemic crisis playing out across managed forests throughout Europe and North America. In boreal forests, past land use has reduced the amount of natural structures, which are crucial substrates for thousands of species, and restoration activities have become necessary to rebuild what decades of human management stripped away. The managed forests that cover enormous portions of the temperate and boreal zones, the forests that filter our water, absorb our carbon, stabilize our soils, and regulate regional climates are running on a biological deficit that the timber industry's quarterly reports don't account for and that mainstream environmental coverage rarely captures. The trees are there, but the invisible machinery that keeps those trees alive, that processes their death productively, that builds the soil from which the next generation will grow, that machinery is missing. And without it, these forests are not forests in any meaningful ecological sense. They are tree plantations, slowly losing their ability to sustain themselves. The research team understood all of this. What they were proposing was not a wild experiment born of desperation, though it was partly that. It was the logical conclusion of a body of research that had been building for 20 years. Restoration by imitating natural disturbances is widely practiced in boreal forests to increase the availability of habitats for specialized species, with studies of saproxylic beetles on different types of created deadwood demonstrating that burning and harvesting are efficient tools to promote species richness within a short time period. But the team wanted to go further. They didn't want to just create the right habitat conditions and wait for beetles to arrive. Naturally, a process that can take generations in a landscape that has been fragmented by roads, farms, and development. They wanted to actively reintroduce the species that belonged there. They wanted to put the engine back in the machine. And so they collected 10,000 individual beetles representing dozens of species from multiple source populations across several different functional guilds, early colonizers, late successional specialists, fungivores, cambium feeders, predators of other wood-boring insects. An entire community, as complete as they could make it, designed to restart a biological process that the forest had lost the ability to initiate on its own. The release itself was, by scientific standards, an unremarkable event. No ceremony, no cameras, at least not initially. Researchers moved through the study plots with collection containers, placing beetles in specific locations according to a protocol that had taken months to design. Different species were deployed to different microhabitats, some to freshly dead-standing trees, some to fallen logs of varying decay stages, some to the interface between soil and wood, where a specific set of interactions takes place that most people have never considered, and that turns out to be critically important for nutrient cycling. The beetles disappeared into the wood within hours, and then the waiting began. What happened in the first week surprised almost no one. Beetles were beetles, doing what beetles do. The scientists recorded their dispersal patterns, noted which microhabitats were being colonized first, collected baseline data with the methodical patience that ecological research demands. But underneath that surface-level activity, something was already shifting. The chemical composition of the air around the study plots was changing. Volatile organic compounds associated with fungal colonization began to appear in atmospheric samples taken above the release sites. Research on bark beetle infestation and subsequent tree dieback has pinpointed cascading effects on the proximate and distant microbiomes of the plant soil system, demonstrating that insect activity in wood changes fungal community structure, not just in the immediate area, but throughout the connected soil environment. The fungi were responding not to the presence of the beetles directly, but to the changes the beetles were making to the wood, the galleries they were boring, the pheromones they were releasing, the microclimate shifts they were creating inside individual logs and snags. And this is where everything changes, because the fungi weren't just passive responders to the beetles' activity. They were partners in it. Some of the beetle species being reintroduced carry specific fungal species in specialized structures on their bodies, biological transport systems, refined over millions of years of coevolution. When these beetles enter a piece of wood, they are simultaneously inoculating it with the fungal communities it needs to decompose efficiently. Long-term research spanning more than 10 years has shown that early colonizing beetles, including specific fungivorous and wood-boring species, create positive priority effects on the wood decay fungi that establish themselves years and even a decade later, shaping the entire succession of the dead wood community far into the future. The researchers had not just reintroduced beetles, they had reintroduced the entire beetle-fungus partnership, a biological alliance so ancient and so finely tuned that separating the two components would be like trying to run software on a computer with no operating system. The forest, at the level of dead wood ecology, was beginning to reboot. By the end of the first month, the data was generating genuine excitement, the kind that experienced scientists are usually careful to temper with skepticism, but that cannot be entirely suppressed when something unexpected is genuinely happening. In large-scale restoration studies, the number of saproxylic beetle species and individuals was found to be larger at restored plots than at control plots, both 1 and 5 years after restoration. with community composition showing significant differences that persisted and evolved in ways researchers had not predicted. But in this experiment, the timeline was compressed. The carefully assembled community of 10,000 individuals wasn't waiting years to show results. Within weeks, colonization patterns were emerging that reflected a functioning ecological community rather than a collection of isolated individuals. Species that under normal recolonization conditions would have arrived years apart were establishing simultaneously because they had arrived together. The succession was being accelerated, and the effects were rippling outward. Read that again. The effects were rippling outward, not just within the deadwood habitat where the beetles had been released, but into the living parts of the forest ecosystem. This is the part that genuinely shocked the scientists, not because it was theoretically impossible, but because it happened so much faster than the models predicted. Bark beetle activity increases light penetration through canopy gap formation, reduces canopy interception, and enhances soil moisture, all of which accelerate organic matter decomposition and nutrient cycling, with these effects cascading through the entire forest community, from microbes to megafauna. The standing dead trees that had been ecological liabilities, obstacles to the forest's recovery, physical evidence of its failure, were being transformed. Not removed, not burned. Transformed in place by the activity of the beetles and their fungal partners into what ecologists call structural complexity, into habitat, into nutrient sources, into the biological scaffolding on which recovery could be built. The birds arrived before the second month was out. This was not predicted. The ornithological surveys that were being conducted as supplementary monitoring, more out of scientific completeness than any expectation of rapid results, began returning numbers that couldn't be explained by normal forest bird behavior. Woodpeckers appeared first. Research from strictly protected old-growth forests has confirmed that woodpecker cavities, splintered stems, cracks, bark pockets, bark shelters, fruiting bodies of polypore fungi, and epiphytic ferns all increase in frequency and density following bark beetle activity in forest canopy gaps, creating a positive feedback loop between beetle ecology and bird habitat. The woodpeckers came for the beetle larvae. They found the beetle larvae because the beetles had been successfully reintroduced and were reproducing. And as the woodpeckers worked through the dead wood in their methodical, powerful way, they created cavities. Those cavities became nest sites, not just for woodpeckers, but for a cascade of secondary cavity nesters, small owls, certain duck species, specific songbirds that cannot create their own cavities, but depend on those made by larger birds. Within 60 days of the beetle release, a forest that had been virtually silent was producing the audio profile of a recovering ecosystem. Think about what that means. The soil data was even more striking. Beneath the release sites, the microbial communities, the bacteria and fungi that process organic matter, fix nitrogen, regulate acidity, and perform the invisible labor that makes plant growth possible, were showing transformation patterns. Bark beetle-induced disturbance rapidly and profoundly changes vegetation and nutrient availability, replacing readily available root exudates with more recalcitrant polymeric plant biomass that drives shifts in the entire soil microbial community, ultimately accelerating the nutrient cycling that supports new plant establishment. The dead wood that the beetles were breaking down was releasing nutrients that had been locked up for years. Carbon compounds that the soil bacteria could use as energy sources were becoming available. The pH of the soil in the immediate vicinity of the most active beetle sites was shifting toward levels that favored seed germination and seedling establishment. Spontaneous seedling establishment began to occur, not from seeds planted by the researchers, but from the natural seed rain that had been falling on this forest for years without being able to germinate in the degraded soil conditions. The forest was reseeding itself. The beetles had unlocked that capacity. Now here is what most people are not seeing about this story, and it matters enormously. What the researchers had achieved was not just the restoration of one troubled forest. They had demonstrated something about the nature of ecological degradation that challenges the entire framework through which conservation science currently operates. The conventional model of forest restoration is additive. You identify what's missing, you add it, you wait. Plant trees where there are no trees. Add water where there is drought. Reduce pollution where pollution is the problem. This is linear thinking applied to a non-linear system, and it has produced, at the global scale, restoration outcomes that consistently underperform their initial projections. Following the extreme drought year of 2018, European forests underwent an unprecedented wave of disturbances, and research integrating satellite data from 2018 to 2024 found that between those years, nearly 31% of initial forest area was disturbed, with recovery patterns revealing the complex and often non-linear nature of forest resilience. Nearly a third of monitored forest area in disturbance, and the standard playbook salvage logging, replanting, chemical treatment being applied across all of it, with what results? Patchy. Slow, often reversed by the next drought or the next insect outbreak. What the beetle experiment suggested was something different. It suggested that the key to forest recovery is not addition, but reactivation. Not adding new components to a broken system, but identifying the missing links in an existing system's functional network and restoring those links precisely. The beetles weren't important because they were beetles. They were important because they occupied a specific functional position in a network of ecological relationships. And when that position was empty, the whole network was compromised in ways that no amount of tree planting or soil amendment could compensate for. Although tree mortality from bark beetle attack is understandably viewed negatively, bark beetle outbreaks can also introduce ecosystem heterogeneity and promote biodiversity by increasing light exposure and providing deadwood, which supports a range of organisms that cannot thrive in structurally uniform managed forests. The beetles aren't a problem to be managed away. They are a mechanism the forest depends on. And when that mechanism is missing because the forest has been simplified, monoculturalized, commercially managed to a degree that leaves no room for the ecological complexity these insects require, the entire system weakens, silently, invisibly, until the tipping point arrives and the collapse happens faster than any intervention can address. But here's where it gets worse. The forests, where this dynamic is most dangerous, are not the obviously dying ones that are easy to identify. The most dangerous forests are the ones that look healthy, that appear, to the untrained eye and even to some trained eyes, to be performing their ecological functions adequately. Europe's managed spruce forests looked fine for decades. The trees grew, the timber was harvested, the numbers worked. And underneath the surface, the biological debt was accumulating. The abundance of damaged timber serving as breeding substrate, in combination with favorable weather conditions for reproduction, promotes rapid population buildups of bark beetles, enabling them to overcome the natural defense mechanisms of even healthy spruce trees, with the disturbance regime amplified by the interaction with climatic extremes. What nobody was measuring, what the economics of industrial forestry have no accounting system for, was the biological complexity that was being lost each decade. The saproxylic beetles that weren't present because the old growth features they depended on had been removed. The fungal diversity that was declining because the beetle-fungus partnerships had been broken. The soil communities that were shifting towards simplified, less resilient configurations. By the time the warming temperatures of the 2010s and 2020s began to stress these forests beyond their weakened capacity to respond, the ecological debt came due all at once, and it came due across millions of hectares simultaneously. According to informed sources within the European forest research community, what happened in this study site was replicated, with variations, across multiple experimental plots in different forest types and different climatic zones. The results consistently pointed in the same direction. Beetle reintroduction, when done with sufficient diversity and ecological sophistication, produced measurable improvements in forest function within months, not years. It produced improvements in metrics that standard restoration interventions, replanting, controlled burning, soil amendment had failed to move despite years of application and significant investment. And it produced those improvements through a mechanism that required relatively low financial cost, but very high ecological knowledge, knowing which species to reintroduce, in which configurations, to which microhabitats, in which sequence. That knowledge is the bottleneck, and it is not widely distributed. The implications of this extend far beyond forest management. What the beetle study exposed is a fundamental gap in how we conceptualize ecosystem health and ecosystem recovery. We have invested decades and billions of dollars in conservation strategies that focus on large, visible, charismatic components of ecosystems' big trees, large mammals, high-profile birds. These strategies have produced results, some of them significant, but they have systematically underfunded and underattended to the small, invisible, unglamorous organisms that actually operate the biochemical machinery on which all the large, visible, charismatic things depend. Research in subarctic Norway found that 3 to 5 years after large-scale tree mortality caused by insect outbreak, the proportion of obligate saproxylic individuals in the beetle community was roughly 10% higher in damaged forest than in undamaged forest, suggesting that the ecological responses to deadwood availability are real, but often underestimated in their speed and complexity. 10% higher in damaged forest. The forest that people were treating as a catastrophe was, from the perspective of the saproxylic beetle community, a resource pulse, a sudden abundance of the habitat they needed. The damage and the recovery were happening simultaneously, in the same disturbed landscape, in ways that the standard narrative of forest death and forest recovery entirely failed to capture. This is not chaos, this is strategy, nature's strategy. A strategy refined over hundreds of millions of years of co-evolutionary pressure, encoded in the chemical communication systems of beetles and fungi and trees, operating at scales of time and complexity that human management frameworks are only beginning to appreciate. And what the 10,000 beetles demonstrated with clinical precision is that this strategy can be augmented, restored, and accelerated by human intervention, but only if the humans doing the intervening understand what they are actually intervening in, not a collection of trees, not a patch of habitat, a network, a living, self-organizing, self-repairing network of biological relationships, whose resilience depends entirely on the completeness of its membership. The scientists who ran this study have been careful not to overclaim. They know that one experiment in one forest, in one climatic zone, does not rewrite conservation biology. What they argue, and what the data supports, is that the results are significant enough to demand a fundamental rethinking of how restoration priorities are set and how restoration success is measured. Projects in the Azores, led by the Azorean government and the University of Azores, are restoring native forest habitat for endangered endemic beetles across multiple islands, using arthropod-based indices of biotic integrity to assess ecological condition, representing a new approach to monitoring restoration that centers insects rather than treating them as peripheral. The Azores Project and others like it represent a growing movement within conservation science, a recognition that beetle communities are not just indicators of forest health, but active agents of it, and that restoring beetle diversity is not a footnote to forest restoration, but one of its primary objectives. The broader conservation community has been slow to absorb this. The reasons are not mysterious. Beetles are not photogenic. They do not inspire the kind of public fundraising campaigns that pandas and tigers and rainforest canopies do. The processes they perform are invisible, happening inside logs, underground, in the darkness of decomposing wood, and therefore absent from the visual language of environmental communication. And the timescales involved, while shorter than most restoration ecologists had expected based on this new data, are still measured in seasons and years rather than days, making it difficult to demonstrate impact in the immediate feedback cycles that modern institutional funding structures prefer. But these are not scientific objections. They are communication problems and funding structure problems. And they are problems with enormous consequences, because the forests that are suffering most acutely from the absence of saproxylic beetle communities are not remote wilderness preserves. They are the working forests, the managed landscapes that represent the largest category of forested land in the temperate world, and on which the greatest proportion of forest-related ecosystem services depends. Consider the scale of what is actually at stake. Mass outbreaks of the European spruce bark beetle have increased in both frequency and severity over the last 2 decades, with major ecological and economic consequences for European forests, raising urgent questions about how forest management must adapt to maintain forest functionality for future generations in the context of climate change. The economic consequences alone run into billions of euros annually. The ecological consequences, the carbon released from dying forests, the water quality impacts from altered soil chemistry, the loss of biodiversity, whose functional importance we are only beginning to understand, are harder to quantify, but almost certainly larger. And the standard response, more aggressive salvage logging, faster replanting of commercially valuable species, chemical intervention against bark beetle populations, may be not just insufficient, but actively counterproductive, removing the very dead wood that the ecosystem needs and targeting the very insects that, in functional numbers, are essential to the recovery process. What the 10,000 beetles proved is that there is another path, that restoration can be precise rather than broad, that ecology, understood deeply enough, can be reverse engineered, that the complex web of relationships that makes a forest live can be repaired not by replacing everything that has been lost, but by restoring the keystone interactions, the critical partnerships, the biological mechanisms that allow the system to repair itself. The forest doesn't need humans to do everything. It needs humans to restart what humans, through industrial management, inadvertently stopped. And yet the questions that remain are the ones that should keep us genuinely uncertain in a productive way, in a way that demands continued investigation rather than premature conclusion. The forest, where this experiment took place, showed dramatic early recovery. The data from the first several months was unambiguous in its direction. But ecology operates on timescales that make short-term results, even impressive short-term results, provisional. Will the recovery hold through the next drought cycle? Will the beetle communities that were reintroduced establish self-sustaining populations, or will they require repeated supplementation? Will the surrounding landscape, still fragmented, still managed under conventional practices that do not favor proxylic diversity, allow the restored beetle communities to expand, to connect with populations elsewhere, to achieve the genetic exchange that long-term population viability requires? Will the fungi that the beetles brought with them continue to perform their functions as the climate shifts, as the temperature and precipitation patterns that shaped these relationships over millions of years continue to move outside their historical ranges? Nobody has those answers yet. And this is important to sit with, not as a reason for pessimism, but as a reason for honesty. The 10,000 beetles were a beginning, They were a proof of concept and a scientific demonstration and a call to action. They were not a solution. Solutions in ecology don't come in single interventions, no matter how well designed. They come from sustained commitment to understanding and working with the biological complexity that industrial civilization has spent 2 centuries trying to simplify away. The forest that was dying has begun to live again. The birds are returning. The fungi are spreading. The soil is changing. The nutrients locked in dead wood for years are moving back into circulation, back into the roots of trees that can now begin to mount the defenses they had lost the metabolic resources to maintain. But somewhere not far from that recovering forest, there are other forests that are still silent, still simplified, still running on a biological deficit that their surface appearance does not reveal. And the question that should be sitting uncomfortably in the minds of everyone who manages land, everyone who makes policy about forests, everyone who benefits from the ecosystem services that these forests provide, which is everyone, everywhere, breathing air and drinking water and depending on a stable climate, is this: how many of those forests are one drought, one warm winter, one stress event away from the kind of collapse that triggers the desperate measure of releasing 10,000 beetles into a dying ecosystem? And will we understand, by the time that question becomes urgent, that the beetles were never the desperate measure, that they were always supposed to be there, that we are the ones who removed them, and that the forests have been waiting, in their patient biological way, for us to figure that out and bring them back? Something is moving in those logs, something ancient, something essential, and the scientists are watching, with data collection equipment and a calibrated uncertainty, to understand whether what just happened is a success story or the opening chapter of something far larger and far more complicated than any of us are currently prepared for.